So hello everyone, a very warm greeting to one and all. My name is Shrigori and I would like to welcome you all to today's webinar on OmicsLogic, NGS, Multi-Omics and Biomedical Data Science. So in this webinar, we will provide a holistic insight into our various training offerings that is designed to empower participants from various skill levels and interests. And these programs span a wide spectrum, catering to diverse learning needs, including introduction to big data bioinformatics, genomic data analysis, transcriptomic data analysis, as well as biomedical data science using Python or R. And before we begin, let me also take a brief moment to introduce you to the team behind the program. We are India and US-based bioinformatics and data science company, and we've had the wonderful opportunity to collaborate and partner with esteemed universities and industries across the globe. To meet the ever-evolving demand of skilled bioinformaticians and data scientists, we have created innovative training and research offerings, including online courses, training programs, workshops, research internships, and fellowships, research services for faculty, scientists, as well as a dedicated online degree program. And through partnerships with coordinators worldwide, we have successfully conducted training and research programs for over 32,000 individuals from 200 countries, conducting specialized program in oncology, infectious diseases, precision medicine, neuroscience, and biomedical data science. And at the core of our training program is the OmicsLogic learning management system, which is a user-friendly platform for students, researchers, and faculties to learn, analyze, and interpret high-throughput data. And here we follow a student-centered learning model where the online programs are divided into different levels based on the background of the participants, that is beginner, intermediate, and advanced. We'll be covering more about it in the upcoming part of today's webinar. And during the course of the program, students will be required to go through multiple resources, including mentor-guided lecture recordings, online quizzes, and assignments, as well as online courses and example projects on topics ranging from introductory bioinformatics, genomics, transcriptomics, metagenomics, epigenomics, data science, that includes bioinformatics using R and Python. And throughout the duration of the training period, we closely monitor and track the learning progress of the students through checking the number of learning points that they have achieved, the number of courses that they have completed, their program progress, and so on. For learning and practicing the analysis, we give two options. For instance, you can learn bioinformatics without any coding at all. So for those interested in opting the non-code pathway, they will be given directions to access the T-Bioinfo platform for bioinformatic processing and analysis of data. And this platform includes demo pipelines as well as data management and analysis infrastructure to run various educational pipelines on bioinformatics. And for those interested in learning to analyze data through coding, we provide the Google Colab notebooks, which will help you develop coding skills while learning popular packages in R and Python to visualize, annotate, and analyze complex patterns in your omics data. And we also get interest from students who want to carry out a research project. So that is why we also conduct a research program that is mentor-guided. And here I wanted to quickly walk you through a few examples on how students have leveraged the learning resources to achieve their academic and professional goals in completing a research program. So we've had a cohort of students from Systems Medicine graduate program at Georgetown University who worked on machine learning projects using Python. And students have also went on to present their research projects in poster-presentation competitions to win exciting cash prizes. And students also join our program to get a research publication done. And students to get a research publication done as well as to showcase as a part of their higher studies. We also provide a business and entrepreneurship track for those students who are interested in the business aspect of it. And here is a quick glimpse into one of the projects that has been worked on by a group of industrial researchers who worked on developing vaginal microbiome testing kit. And students have also worked on it. So we'll be exploring all of these on our platform on the OmicsLogic. So without further delay, I'll quickly take you to the OmicsLogic Learn platform. So here is the link. So can I get a quick response in the chat box? How many participants have already created an account on the portal? Do you have a prior account? If so, I can directly start with the navigation part. Or if we
Yes,
have,
I have an account and I already take one course.
yes, so maybe you were in the workshop with us. What about the rest of the participants? Can you put it in the chat box? You can put a yes if you have already created an account. Okay, we have one participant, Yukta. Okay, all right. I guess a few of the participants don't have an account. So I'll quickly walk you through the process of signing up first and then take you to the part. So I'm sharing the link in the chat box once again. Please visit this link and you will be able to see such an interface. All right. Now, for those of you who have not created an account, click on this create an account option. And here you will be asked to enter your name, such as your name, email ID, provide a password, and few other details such as your educational background and your organization. And you can directly create an account. Now, for those of you who already have an existing account, you can simply click on the login now option, enter your existing email ID and password, and directly click on sign in. And I'll wait for a couple of seconds for those participants. Please put a yes in the chat box if you are good to proceed. Then I can walk you through the next step. All right, perfect. Yukta has done. If you encounter any technical issues, please feel free to let us know. And technical team can look into in case you want to recover your old account or any such issues. All right, we do have a few responses in the chat box that we are good to proceed. If at all, at a later stage, if you have any queries, then we can take it up during the end of the webinar during the Q&A part. All right. So with that, I'll proceed with the next step on logging in. Once you sign in, the very first interface that you see is the courses page. So here, the first step is to complete setting up your profile. Here on the blue bar on the top, it will display your name. Click on that and it will redirect you to your profile settings. My network is a little bit slow today. Now here you will see the profile button, click on that. And here you can update your profile features such as make sure that your name is in proper capitalization and that there is no spelling error. Because when you complete any coursework on the platform, the certificates will be automatically generated. And this is the name that will be printed on your certificate. So make sure that this is done in proper manner. Add a brief bio about yourself, about your education background and your research interests. And you can add a profile image and link to your social media handles. This could be your LinkedIn, Facebook, or OrcID, or Instagram, or Twitter. And once all of this is done, this should be showing profile person 100%. Now, in today's webinar, we'll be covering mainly about the training programs. So this is the link that is when you head over to the blue bar on the top and you hover your cursor over the program, you will see the training program option. So we'll walk you through each of these training programs. So no need to feel overwhelmed that is anyone trying to speak? Okay, I guess it was an error, no problem. All right, so this is the main link to see the various collection of training programs that we are offering. So now I'd like to invite Sonalika Ray, the research scientist and the program mentor at OmicsLogic, who's the expert and who has developed all these training programs, to talk more about these program features and their highlights and the various learning resources. So over to you, Sonalika.
Thank you so much, Shrigori. Can you please confirm if my screen is visible?
Yes, we can see your screen.
Great. A very warm good evening, everyone, or good morning or afternoon in whichever part of the world you are. I'm Sonalika Ray, and I'll be today talking about the different courses and the training programs on the OmicsLogic Learn portal that you can leverage and learn from. But before I start taking you through the courses and the programs, I would first of all like to know your background. You're from the audience that we have is from which all different fields and which all different level of education do you have. So if in the chat you could please write your level of education and your field. For example, if you have done your bachelor's in bioinformatics, simply type in the chat bachelor's bioinformatics or biotechnology or biochemistry, whatever it is, so that I know who all have joined us today. And also, I would like to hear from you about your research interest. So I can see we have two students from biotechnology, one from biomedical genetics and bachelor's in bioinformatics. Okay, Yukta, thank you. I would like to hear from everyone. Let's keep this session more interactive. I know you all have joined because you all have several questions and also interests to get started with a specific training program and you would like to learn about it. So let's make the most out of this webinar and clarify all your questions that you might have. I would like to hear from everyone about their fields and also those who have told me about their fields, please let me know about your research interest. What brings you here today? If you're interested to learn more about genomics, then type genomics, or if you're interested to learn about transcriptomics, then type that. Vishal says genomics. Okay, we are mostly joined by master's students in either bioinformatics or genetics or biotechnology. We have one student from physiology. That's interesting. Only two students have responded. I can see that we are joined by around 30 plus participants, but I can only see few responses in the chat. I would appreciate if all of you can respond in the chat so that when I talk about these different courses and programs, I can address to your interest, your research interest that you want to learn from this portal. Maria says she is interested to learn about bioinformatics. We are also joined by a PhD student in microbiology. Yukta is interested in omics studies overall. Okay, metagenomics, transcriptomics, and more. Okay. We are also joined by a doctorate in veterinary medicine, research interest, biomedical research, and transcriptomics, metagenomics. All right. How many of you are familiar with the OmicsLogic Learn portal and already have explored the courses on the portal and the different training programs? If yes, then please raise your hands. And if not, then do not raise your hand. Balkis is interested in learning genomics and transcriptomics. Most of the interests are around either starting with the field of bioinformatics or diving deep into genomics or transcriptomics. Yukta says she has gone through some of the courses. All right. Sumaya says yes. Sumaya, if I'm not wrong, are you the same student who had joined us in the workshops as well? If yes, then please put a yes so that I know we are joined by same Sumaya. All right. Good to have you back with us. I have one response by the phone Android, Blue Droid. Can you please rename yourself? Okay, we are joined by pathology students as well. Nice. Okay, so let's get started. This is the portal. Sri Gauri has already given you a good overview of what the portal is all about. There are three major aspects that I'll be talking about today. The first one are the courses. The next sort of training programs that you can go through are the training programs, which consist of lectures and recordings that you can go through, hands-on analysis that you can go through. And the third aspect is of research, where you can work on a research project, because just learning about a specific concept and not applying to a research question leaves it incomplete. So if you learn something and then also you apply it to a research question, it gives you a good understanding of the concept. Okay, so on the OmicsLogic Learn portal, there's a tab of courses. Now, if you click on the courses tab, This is what appears on the screen. There are several different course categories. To get started, you have bioinformatics, genomics, transcriptomics, machine learning, Python, R, metagenomics, cheminformatics, single cell, and epigenomics. And for a beginner to get started, who is from a different field, let's say microbiology or biotechnology, and does not have a good understanding of what bioinformatics is all about, and if you're a bachelor student and you're pretty new to this field, then I'll recommend that you should get started with the coursework Bioinformatics. The course Introduction to Bioinformatics is absolutely free. It's open for everyone because this is an introductory coursework and this is for you to get an overall understanding of what the field is all about and what different fields does it include. When I say bioinformatics, we in the initial lessons we talk about what bioinformatics is, how it came into existence, how did it evolve, etc., etc. And then we start discussing about the different omics fields like genomics, transcriptomics, metagenomics, and then we talk about the other specifics in a coursework that is called as bytes and molecules that I'll just open and show you, where we also introduce you to different research specialization tracks. I'll definitely discuss about different research specialization tracks as well. But before that, I just want to show you the structure of the coursework. Now, let's say if you are a student or a researcher or a working professional and want to gain a skill set and do not have time to attend live lectures, you can take up the courses that you can go through at your own pace. The courses are pretty much like a book, you can say, and different lessons are chapters here. So if you open up a lesson, you would get some text to read, some images to visually make you understand the concept, and then you have some videos embedded in the lecture itself, in the lesson itself, that you have to watch. And then towards the end, you have some quiz for completion of the lesson, and that makes the lesson complete. So like this, you can make steady progress at your own pace whenever you get time, and you get to learn a new concept. For every lesson that you complete, you get some points that adds up to your profile, and in the profile itself, you'll get the certificate as well. I'll talk about that towards the end of my explanation of courses and programs to you. Now, after bioinformatics, I just mentioned about a course called Bytes and Molecules. Now, the specific course Bytes and Molecules, it introduces you to the different fields of omics, and it also introduces you to the different research specialization tracks. Now, what do I mean by different research specialization tracks? There's often a confusion among students who get started exploring the field of omics. For example, you learn about a specific omics field, be it genomics. The next question is, what type of research questions can you address through the genomic data? So we have classified the research specialization tracks into 6 different fields that you can opt for and explore. It could be some other field as well. It depends on your research interest, your research question, but mostly it comes under these 6 research specialization tracks, which includes oncology, neurosciences, precision medicine, infectious diseases. One is space omics, and one that's not mentioned here is also the field of agriculture, where you can explore the omics data and unravel some insights out of it. So that is what the Bytes and Molecules course offers. Now, let's say you have you just go through these introductory courses and you got started with these courses, and now you understand what different omics fields are out there and what different research tracks are there where you can apply your knowledge of omics data analysis. The next step is to get a good hands-on with a specific sort of field. So some of you mentioned that your interest is genomics. When you talk about genomics, I'll just click on genomics, I'll click on explore, and then I'll get a coursework on genomics, and then I can see some projects on genomics as well. What are these projects? Coursework is definitely something that will help you get the concepts clear. and also get a good hands-on with the practicals. And then these projects are just example projects for you to understand how different sort of analysis that you learn from this coursework, how you can apply that analysis to a research question. So we have included some research projects here where we address different research questions. So when we talk about genomics, there are some topics that we have covered here. If I talk broadly, the first one is variant calling. So let's say you get this genomic data from a sequencer, and this genomic data can be in the form of FASTA files or FASTQ files, mostly FASTQ files. Once you have these files, you need to trim the adapters, and then you need to remove the duplicates, do some pre-processing, and then you need to understand how to check the quality of the reads that you got from the sequencing experiment. So how do you perform quality control? And then how do you perform alignment? That is, how do you align reads to a reference genome? Then how do you look at the variants? That is, when you align the reads to a reference genome, the bases are matched upon, and if there's a mismatch, it could be a variant based on how many mismatches at the same site are observed for different samples that you take. And very broadly, I'm explaining you what all different topics we are covering. So one of the major topics that we have talked about here is, once you get the FASTQ files, what do you do with that? And the first thing is variant calling, where you look for different sorts of variants that are present in your data, in your samples. All right, once you have these variants, what's next? You get the variants, then you filter the variants that to understand which variants actually statistically significant and make sense. And then you try to annotate those variants based on the ACMG guidelines. That's a classification system that has been assigned to variant annotation to understand if a variant is pathogenic, likely pathogenic, or benign, or likely benign, or non-significant. So that is one sort of analysis that you can learn in this coursework. What I said: variant calling, variant filtering, variant annotation. The next sort of analysis that you can learn from this coursework is phylogenetic analysis, and where you will learn how you can align multiple sequences and then construct a phylogenetic tree and then perform your evolutionary studies on it. And then you can also learn CNV analysis and mutability analysis from this coursework, right? Now, there's a difference between a coursework and a training program that I want to highlight here. So I just talked about introduction to bioinformatics, and this is the second coursework that I talked about, that is genomics. Now, what happens generally if there's a student who has just started to learn about a specific field, you might not be able to catch up with the concepts just by reading through it and understanding the practical hands-on. So that's why we have put together some training programs. Now, what are training programs? Training programs are just like online training sessions that we have, but instead of having live sessions, we have them recorded, and we have put those recorded lectures here that you can access at your pace whenever you have time, right? So how do you get to a training program? On the OmicsLogic Learn website itself, I explain you what are courses. If you go to programs, you get the category as training program, workshop program, and diploma. We run a diploma program, a PG diploma program with IIT Jodhpur. I'll talk about it towards the end of my discussion of programs with you. Let's now focus our discussion on training programs. So if you click on training programs, you will get the entire list of training programs. If you are looking for something specific, simply type it up and you will get the training programs available for the keyword that you type. Now, if I click on Introduction to Big Data Bioinformatics program, and if you scroll down, you can see the entire material for this training program. I can see the entire training material because I have logged in and I'm a part of this program. If you are new on the website and you have not yet purchased a training program, you will not be able to view the session videos, obviously, but you will get an idea of what all topics are covered and what all subtopics are covered under each and every session, so that you get an idea and then you can register. Shubhagna will help you understand how you can register yourself for the program. I'll pass on the stage to her after some time. Okay, so here, what is there under each and every session? On the training program page, we have divided your learning into 3 different levels: beginner, intermediate, and then there's an advanced level. Beginner is obviously to get you started, and here you have these lectures put together for each and every session. Each session might have 1, 2, or 3 videos discussing about some different topics. And here we also introduce the topic, we discuss about the concepts, and we also perform practical hands-on. And along with each lecture, you will get an associated lesson. This associated lesson is the lesson from the coursework of Introduction to Bioinformatics, the one that I just showed you. And then you have some additional reading material. These additional reading materials could be either research publications or some articles from the internet itself, or it could be some animated videos that we would like you to watch. We do not focus on explaining the concept of sequencing in detail. We just focus on the dry lab aspect. So for wet lab aspect in the lessons as well, we have referred to some nice animated sequencing videos that you can watch and understand how the process works. All right, so for the sessions, you will find all of the associated lessons and the resources that you have to go through. And wherever in the coursework we perform any practical hands-on, we have mentioned the link of the demo pipeline on the Bioinfo server. I'll explain you what this server is all about. And also, if we are performing the analysis on an open source tool, or if we are performing the analysis using R or Python, then you will get the Google Colab link as well. The training programs and the courses, they focus on 3 different aspects of analysis. How can you perform the analysis? Using open source tools and softwares, because everyone is not a coder here. And for the coders, how to perform the analysis using R or Python. Or if you want to learn coding, I'll show you R and Python courses from where you can learn the analysis, get started, and then refer to these collabs to perform the analysis. And the third way is an automated way where you run the pipelines with your data and you get the results in the required format, right? So mix of 3 different ways you will learn to perform different sorts of analysis. So that is how these training programs are arranged. Now it's your wish. If you want to, based on the topics that are mentioned, you can pick whichever course you would like to get started with. Now there's one general question that we usually get if a student should take the coursework or the training program. If you are a complete newbie and you have to get started with the field of genomics, or let's say with the field of bioinformatics, I'm talking about the bioinformatics program here, so I'll focus my discussion on bioinformatics. If you just want to get started with bioinformatics, then we'll recommend that you start with the training program. In the training program, you would have to go through the coursework as well to supplement your knowledge. But if you start with the coursework, then you are on your own. As in, you have the material that is put together and you have to go through it. And if you're able to understand it, good for you. If you need assistance, then you can reach out to us. In the training programs, you get sessions with the mentor. So you have these recorded lectures that you can watch, and then you also have some live Q&A sessions that help you clarify your doubts with the mentor. But if you have taken up the courses, you don't get the access to the live Q&A sessions with the mentor, and that is one of the drawbacks. But if you think that you want to just brush up the concepts and you'll be okay with not having the live Q&A sessions with the mentor, then definitely go ahead with the course subscription. Okay, so that is about the introduction to bioinformatics training program and the genomics coursework that I discussed. Now, for the genomics program, we also have this program put together, that is genomic data analysis training program. And it also has the 3 levels: beginner, intermediate, advanced. And in the similar manner, you have the session videos put together, the associated lessons, and you also have some additional material that you can go through to supplement your knowledge. And also you would have the link of the tools that we would refer for analysis. And as I mentioned, we are covering 4 different analysis here: variant calling, filtering, annotation, phylogenetic analysis, CNP analysis. And also we have introduced Jivas here. That's pretty much that's what we have covered in the analysis in the training program. And here we have also introduced how you can perform the analysis using R. For example, you learn about a concept and then you learn how to perform the phylogenetic analysis, and then you learn how to perform the same sort of analysis in R. Okay, so that is about introduction to bioinformatics and genomics. I'll take a pause here. If you have any questions, please put in the chat so that I can address. And if not, then I'll move ahead and discuss about the other training programs, which are Transcriptomics and Metagenomics. Are there any questions? I'll pause for a second. All right. Celine says, all okay, just one response. So I will now proceed ahead with another coursework that is on transcriptomics. My major objective for this webinar is to explain you what all topics we are covering in the training program. And if you think you want to learn the same, then you are most welcome to join the training program with us. Shubhanga will explain you how you can enroll. And let's say if you have a specific interest and I did not mention that topic, then you can write us an email and we can help you with the analysis on one-on-one consultation as well, or one-on-one live sessions or tutoring sessions as well. Now, it also might not be feasible for me to talk about each and every subtopic, keeping in mind the time of the webinar. So I'll be just giving you a broad overview. For the details, please feel free to go to the program page and read through the subtopics that are mentioned here. Now let's talk about another training program, that is Transcriptomic Data Analysis. I opened the coursework Transcriptomics and I have opened the training program already. So let me quickly open this Transcriptomics coursework. Now, what do we cover in the Transcriptomics training program? The training program and the coursework are in sync to each other. So I'll just explain the flow that we are talking about here. So there are different aspects again that we discuss here. The open repository.
Hi, Sonalika. Can I interrupt?
Yes, sir.
Yeah, it would be good if you can log out and then show, because that will show the curriculum, not the...
All right, sir. So for opening the Transcriptomics training program page, I'll go to the programs, click on the training programs, and here is the Transcriptomics program page. The view that was visible before was the one where I had logged in, and because I'm a part of this training program, I could see the content over there. That is the videos that are uploaded and the material that is out there, which includes the links, the additional material, and the pipeline links as well. I'll just explain you what topics we are covering here, and then I'll take you to the resources and show you what to be covered. Now, coming back to what topics are we discussing here in the Transcriptomics training program. So we start from the very basics. So this program is ideal for the ones who want to start with transcriptomics. It's not important for you to have an understanding of transcriptomics before you start with this training program. You just need to be a biology student and be able to understand the basic concepts of biology, what is a central dogma, and the basic terminologies. So we start off in the program by discussing the different types of and the techniques of sequencing platforms. What are the different techniques of sequencing? What are the different methodologies of sequencing? And what type of data do you get from a sequencing experiment? Once you have the data, what do you do with it? Once you have the data, how do you pre-process it? How do you perform quality control on it? And how do you get a gene expression table out of the FASTQ finds that you get from a sequencing experiment? Now, generally, there are two sorts of students or researchers who want to join the transcriptomics training program. Either they're interested to learn how to perform the analysis. In that case, they don't have the data with them, but they just want to learn the analysis. So it's very important for them to also learn where in the different repositories you can get the data. And when we talk about transcriptomic data analysis, there are different techniques through which this data is generated. So you also need to learn about the different types of data that you could get. What do I mean by that? You could either be working with microarray data or high throughput sequencing data. So we also discuss about the different repositories and how do you download these different types of data that you can work with to analyze the transcriptomics. And if you're a researcher who has generated data in your own laboratory, then you might be having the FASTQ files and you would be interested to generate the gene expression table. So we'll discuss the entire process. How do you do that? Now, once you have the gene expression table in hand, what's next? Once you have the table, it's important that you clean the data. You prepare it for analysis. Either you perform normalization or that's quantile normalization or log transformation on it to generate a PCA plot to perform dimensional anti-reduction and understand what are the outliers and what is the sample variance of the samples that you have in your data. And also we will learn here how you can explore your data to understand what is the sample variance, like how similar your samples are or how distinct your samples are in the data set that you are working with. So we go stepwise and we try to understand what to do, how to do, and what's being done and why. So that are the major questions that we'll be addressing through each and every step. Now, once you have cleaned your data, once you have removed your outliers, the next thing is you perform differential gene expression analysis. Now, for differential gene expression analysis, we will discuss three parts here. The first one is the automated path, where you get the clean data, you run the pipeline on the Bioinfo server, and then you get these results that you get to interpret. Or the other path, that is, if you don't want to go through the automated way, but if you want to perform the analysis using open source tools or softwares on your own, then we'll also introduce you to these different tools that you can use and perform the analysis. And then we will also discuss about how you can perform all the same steps of analysis using R. So that is what is covered in the intermediate and the advanced level, where we discuss how the same sort of analysis can be performed in R. From differential gene expression analysis to function characterization. That is, once you get to know what are the differentially expressed genes present in your data set, the next thing is to understand what's happening with those genes. Which pathways does it affect? Which biological processes is it a part of? Which functions is it a part of? Where is it present in the cell? And which other genes does it interact with? So all of that comes under function characterization. One way is that you do it using the pipelines or through the open source tools and softwares, or you do through R. We also introduce you the concept of machine learning in this training program, where we discuss about unsupervised analysis and supervised analysis for RNA-seq data. Why? If you get to learn about the unsupervised analysis using R, you will be able to create these PCA plots and look for the samples, similarities, understand the variance, understand how your samples are distinct. And the supervised analysis will help you extract the differentially expressed genes, or I should say extract the features out of your data set. If you are new to machine learning, you need not worry. We again here start from the very basics. Each and every topic that we start off in the training program to talk about is from its introduction. If you know about the concept, you can quickly revise your concepts and proceed ahead. Otherwise, the introduction will help you build on the basics. And that is what we talk about in the Transcriptomic Data Analysis Training Program. And towards the end of this training program, we also introduce single cell transcriptomic data analysis, just to get you started with single cell transcriptomics. There are two specialized fields that's being talked about these days a lot by the researchers. One is single cell transcriptomic data analysis, and the another one is spatial transcriptomics. We also plan to add spatial transcriptomics in our training program. For now, we have added the single cell to give you a good glimpse of both the types of analysis along with machine learning and R in the training program. This training program is a power pack program, I'll say, where we cover all the important topics that are required for you to perform an end-to-end RNA-seq analysis. There's this last session in all the advanced training programs that says submit your research proposal. So the students who take an advanced level of the training programs, they are given a chance to submit their research proposal, as in you complete the entire training program, and based on whatever you learned through the program, towards the end of the program, you submit a research proposal, and we'll review your research proposal, and then you can join the research fellowship training program or the research fellowship program, where you get to work on a research project that you can either present in a poster presentation competition, or you can get a conference paper out of it, or a research publication, whatever your goal is. So towards the end of each and every training program, you will find this option to submit your research proposal once you have completed the advanced level of training. Now, there are students who have not worked on a research proposal before, or have not worked on a research project as well. There are many bachelor students who take these programs to learn a concept or get started with the field. So for them, at this stage where it says submit your research proposal, we also provide lectures. We have these recordings that we have put together for this step, where you get to learn how to review literature, how to create a research proposal, and what is the right way of creating one, and how do you work on a project, and what are the steps that you should follow, and which are the resources that you can refer to. All right. So that was about the Transcriptomics training program. And in the training program, you will get the links to the softwares, the tools, the packages that we would be using, the pipelines as well. And in the program, for each and every session, you have the lessons linked to the Transcriptomics coursework that you can refer to, that you have to refer to for an in-depth understanding of the lecture or the concept. For example, I'll just open one of these lessons here and show you what would you have. I'll have to log in. I'll maybe give you a glimpse of the coursework towards the end, once I'm done discussing all the training programs. Okay. And in the coursework as well, we cover all the topics from the very beginning towards the to the end. And also we have introduced machine learning for RNA-seq analysis in the Transcriptomics coursework itself. And for single cell data analysis, there's another course that is on single cell data analysis. And even that course and the lessons of that course are linked in the training program itself. You need not go to any different place or any different resource when you get started. You just have to follow this training program, and if you click on the resource, you'll be automatically redirected to the specific lesson that you have to complete for that session. Yes, Nishant, you will get a recording for this session later as well. Augustina, I'll get to your question just in a while. Now, coming to the next training program that I want to discuss about is metagenomic data analysis. One of the participants in the chat mentioned that he or she is interested in learning metagenomic data analysis. So when you talk about metagenomic data analysis, there are two kinds of analysis that are very important to learn about: data 2 and kind. So we cover both of these different types of analysis in the metagenomics training program. But let's say you are a newbie and you're just getting started with this field. So we first of all talk about what the field of metagenomics is all about. What are the different sequencing techniques? How to generate metagenomic data? What are the different open source databases to get the metagenomic data to work on? Obviously, if you're a student, you might not have the access to this data. So we explore these databases, we extract the data from there, and then we perform the analysis on it. So we'll discuss how to extract the data from these different databases. Then we'll talk about the different tools that are available for metagenomic data processing. And then we start performing the analysis. The two major analyses that we would be performing is data 2 and kind 2. For those who have, or for those who are learning about this field from me for the first time, metagenomic analysis is generally trying to understand. So I'll give you an example here. If you have a handful of soil and you want to understand which microbial communities are present in that handful of soil. So you perform a metagenomic, you get the microbiome from there, you perform a sequencing, you get some sequences from there, and then you would run through these analysis. Obviously, you will do a quality control first to understand the quality of the reads. We'll discuss each and every step in the training program. And then you perform these analysis of data 2 and kind 2, where you get to know which communities are present and what is the abundance of these communities. As in, when we talk about microbial communities, we will talk here about their taxonomy. That is, which kingdom they belong to, or which phylum, and then the family, order, genus, species. In that order, you get to know about the taxonomy classification of the microbiome that is present in this handful of soil, as an example that I told you. Or if you're interested to see in a diseased patient, you're interested to see how the microbiome dysbiosis happens and which are the communities which are in more abundance or less abundant. And then you want to compare it with the healthy sort of individuals. So you can take the samples from the healthy individuals, from the patients, and then compare the microbiome composition of these two groups and then understand which community is in abundance or less in abundance. I'll not get into more details of it because we might have some non-microbiologists here, so I'll not confuse them. I just wanted to give you an idea of what are we trying to do in this training program. We talk about these different types of analysis. We talk about the steps involved. We talk about the tools that are being used, what parameters are considered and why. And most importantly, how do you interpret the results? It's pretty easy to follow something, to follow the steps and get the results. But how do you make sense of those results? To extract insights from it that you can report in your research work. That's one of the most important things that we talk about in our training programs. And in the advanced level, we talk about functional metagenomics. That's being talked about a lot these days, where researchers are trying to understand how a dysbiosis can affect a specific molecular function in a human body, let's say, if the human body is a subject of interest. And we'll also talk about different sorts of case studies here. And we'll talk about metagenomics-based personalized medicine. And then we dive into R, how metagenomic analysis can be performed using R, and how different types of analysis, as in taxonomy, classification, dimensionality reduction, phylogenetic analysis, could be performed using different packages in R for metagenomic data. And same as I told you before, towards the end, you get a chance to submit your research proposal that would be reviewed by our team. Now, just like the other training programs, you also have a course that is associated with metagenomics. And for metagenomics, when you get to log into the page, you get to see the links associated, additional reading material, the link of the tools, the link of the packages, and the resources that you have to follow for this training program. Now, coming to the training programs on R and Python. Now, there are, again, two categories of students. Ones who want to get started with R or Python for omics data analysis, and others who want to learn a specific type of analysis and are well equipped with the basics of R and Python. So let's say you are a newbie in the field and you just want to get started with R or Python. I'll quickly log in and I want to show you two major courses that are associated with R and Python courses, and how do you go through each one of them if you are a newbie or if you want to learn a specific type of analysis. Just give me a second, let me log in. All right, I'm here. So for those who just want to get started, you will find two courses on R, and there are two courses on Python as well that I just showed you. The first course is for beginners. Now, some biologists do not have a computing subject in their field. It could happen that if you're not from a bioinformatics field, but a core biology field, you might not have a computing subject. You might not have had a computing subject ever. Because of that, you don't know the basics of a computing language. So that is why in the first course, we will talk about the basics of a computing language. We'll first introduce you to R and R Studio, how to download them in your system from very basic. Then we talk about different omics data, because using R or Python, you can analyze different omics data. So if you're getting started with R directly, it's important for you to learn about these omics data types. It's not important that you have to be proficient in each and every omics analysis. Get started with one and then keep on adding the other skill set, the other analysis skill set as you proceed ahead. Then you learn the basics of a computing language, how to import data, how to manipulate data, how to explore data, how to create different visualizations, and you will be introduced to different R packages as well. And you will be introduced to Bioconductor. Bioconductor has amazing packages and tools available for bioinformatics analysis, for omics analysis. That's why it's very important for you to know what Bioconductor is all about and what sort of analysis is possible with Bioconductor. Once you have a good background set for getting started with R, then comes the second course, that is R course 2. Now, it is recommended when you get started with the coursework in R, you take up the training program. Why? Because the training program, you have these lectures that will help you understand the code blocks and practice them. So the order of the courses, the order of the lessons in the course that I am showing you, the same order is followed in the training program as well, where we first talk about how to install R in R Studio. We talk about data structures and data types in R, how to import data, how to manipulate data, how to visualize data with ggplot2, and how to perform dimensionality reduction. And then we also talk about how to select features and perform statistical analysis in R. I think there's some loading issue here. I can't load the training program. Okay, now going back to our course 2, the specific course is a more specialized course where you get to learn how to process and visualize data, how to perform a data visualization. Now, one of the most important things when you perform any sort of analysis is your capability to show your results through visualizations. So it's very important for you to learn how you can create these different sorts of plots, different sorts of visualizations using these different tools that are available. So you get to practice these different visualizations, be it bar plot, box plot, histogram, or a scatter plot through different tools in R. And then you'll also get to learn how to perform statistical analysis and RNA-seq analysis using R. And then you have some lessons around machine learning. So that is what we talk about in biomedical data science using R program. And then you have the program on biomedical data science using Python. For Python, just like R, you have two courses. The first course is to get you started, and the other course includes more specialized topics. I'll open both of these courses. Okay, so in the first course, we'll discuss the basics of Python packages, how these packages could be used for omics data analysis, and then how you can perform some basic basic tasks with Python. And then in the second coursework, that is a more of an advanced coursework, you get to learn how you can visualize data using Python, how you can perform descriptive statistics using Python. And as you know, Python is mostly used to build machine learning models. So here we talk mostly about machine learning, how to perform different tasks of machine learning using Python. And in a similar way, we follow the topics in the coursework, in the training program as well. I'll just take you to the training program. If you have any questions, please feel free to put it in the chat. Once I'm done explaining these programs, I'll attend to your questions. So you open the biomedical data science using Python training program, and the same order of lessons that I just mentioned in the coursework are being explained here as well. And for those who are new to machine learning, we first introduce machine learning in the training program, and then we discuss about different sorts of analysis that can be done using machine learning, how to build a predictive model, how to perform unsupervised analysis, how to perform supervised analysis using different algorithms, and how do you interpret your data. And then towards the end, we also discuss about regression and generalized linear models. And we also introduce deep learning in this training program. And towards the end, you get a chance to submit your research proposal. So that is mostly what we cover in the Python training program as well. Minahal for financial aid, Shubhanga will help. Okay, so that was all for the different training programs that I wanted to discuss. Now, these training programs, beginner, intermediate, and advanced, require a month, 2 months, and 3 months respectively for you to learn a specific skill set in depth. For you to learn a skill set, and the skill set that you will learn would help you complete a project end to end as well. So that was all about the training programs. But let us say you don't have much time to allocate in learning a specific skill set, but you want to learn it quickly. Maybe if you have a project that you want to work on, or the demand as per your interest. So for that, we have these workshops that you can join. And there are these different workshops on introduction to RNA-seq data analysis or RNA-seq using R, an introduction to Metabolomics, introduction to R programming that you can join and work on. So these workshops are generally for a week. We have 3 sessions on alternate days, and you're given an access of 2 weeks. that is 14 days to complete the analysis and the hands-on that we discussed in the 3 days of the live sessions. So that is about the workshops. If you have any question regarding the training program or the workshop, please put it in the chat and I'll be able to address. Augustina is asking, is it possible for the course to learn about novo assembly, de novo assembly of the genome of... So, Augustina, the specific training program won't help you understand how to perform de novo assembly on a specific strain. We do discuss about de novo assembly, but because not everyone has the system capacity to perform de novo assembly, that is why it's not being taught in the hands-on practical though, but we talk about the concept. If you're interested to perform de novo assembly, please write us an email and we'll be able to guide you how you can perform one on the stream that you are interested to work with. Thank you, Sri Gauri, for putting up the mail. Augustina, you can write the mail. You can write an email to us on communication at OmicsLogic.com as Sri Gauri has put in the chat. Okay. So far I discussed about these two different types of training programs. Now, the last one that I want to talk about is the research program. So once you have completed a specific training, be it genomic, be it transcriptomic or metagenomic, or be it cheminformatics or metabolomics, the next step is if you want to work on a research project, how do you get started? So I would now like to quickly discuss about this program that is called as a research program. And I would like to tell you how this program helps you work on a research project. And towards the end of the project, what will be the outcome? So once you complete a training program, you submit a research proposal towards the end of this training program. It gets reviewed by our team, and then I mentioned that you get started with the research project that you would like to work on. Now, you can join the research program either for 2 months, 3 months, or 6 months. It depends on the purpose for which you are joining the research program. Some students join the research program to complete a dissertation with us. If you're a bachelor student or a master's student, every college has this norm that you have to work on a dissertation project and submit it to your university to complete your degree, right? So if you want to work on a dissertation, you can enroll in the research program, work on your dissertation. If it's a mini project, 2 months would be enough. If it's a major project, 3 months would be enough. The outcome of a 2 months mini project or a 3 months project could be either having a project report or a conference paper, or it could be also being able to complete a project to present a poster in a poster presentation competition. And if you want to publish a research work, it's required that you review literature because for a research publication, more work is required, and that's why you need at least 6 months to complete on a meaningful research project and find something novel, find some novel insights from your research that you are able to publish. So that is how you can work on a research project. Now, how a research fellowship program or a research program works. In this specific program, we have one-on-one sessions with the students. And in these one-on-one sessions, we tell the student, we discuss what the research question or the interest of the student is. And we assign tasks to the student to work on a research project. The research project can be proposed by the student based on their interest, or if they don't have a specific research question that they want to work on, we already have some predefined research problems for you to work on. So when a student joins, we tell them what different research problems we have defined, and based on their interest, they pick one and we get started. And in these one-on-one meetings, we tell the student what they have to work on. We discuss if they have any questions or queries. We clarify their queries and discuss their progress and give them the next steps that they have to work on. So it's that way we meet, we discuss, we work on the project, we clarify the questions, and we get started with the next steps. So that's how a research program works. And with that was all that I wanted to discuss with you. If you have any questions, please put in the chat for me to address, or you can also unmute yourself and ask any questions that you have. And if not now, then I would pass on the stage to Shubhangna, where she can help you understand how you can register for the training programs, as most of you are waiting to understand how to register for the program that you are interested to join. Shubhangna, over to you.
Thank you, Sonalika, for this wonderful explanation. So if there are any questions, participants can put their questions in chat box. So moving on towards the registration process, I share my screen and give you a brief overview on how to register for the participation. So we have these training programs. When you log into OmicsLogic portal, go to the training programs tab. And here you can see different training programs like biomedical data science using Python, introduction, or based on your interest, you can select the training program. As most of the students today were interested in genomics, so I'll show you with example of genomics program only. So you simply have to click on genomics training program. And when you scroll down, you will see different levels. So you can see beginner, intermediate, and advanced level. So with beginner level, you will have access to 1 month. With intermediate, you will have access to 2 months. And with advanced one, you will have access to 3 months. Also, intermediate will cover sessions from beginner. Similarly, advanced will cover sessions from beginner and intermediate. Now you can check the syllabus here when you drop down. And for the enrollment part, you simply have to click on Buy Now option. As you all know, we have notified you all by email as well. We are offering a 50% discount right now on all these training programs. So that is the financial assistance we are providing to all the students and participants who have enrolled, especially in this webinar. So what you can do is you can simply write Omics 50. That is the discount code for all the students. You have to click on apply, and once your coupon is applicable, you have to tick mark this box, and as you can see, the discounted price will be mentioned here. So if you are from India, you can select Razor Pay option, and if you are from outside India, you can select PayPal, or if you want any alternate options, we can provide you with that as well. Like we have bank transfers, UPI option as well. With the Razor Pay itself, you will be further proceeded towards the UPI option. So as you can see, I have shown you the Indian pricing. You can simply select your region and select USD pricing as well. So when you click on enroll now, you will see all the pricing that are displayed here will be coming in dollars. So simply you can select one of the level. For example, I'll be selecting the advanced level right now. So when I paste the code Omics 50, it will be applicable and I get a discount here. The discounted amount will be displayed on my screen. So as you can see, you are basically receiving advanced level and the pricing of less than intermediate. So it's a good deal going on right now. And all these discounts are applicable till 20th of July. So you have 3 days for enrollment. You can let us know if you are facing any difficulty with this. You can also reach out to our team for this. Also, as Sonalika talked about workshop program, the process for workshop enrollment will be the same. You simply have to go on any workshop, as I'm already enrolled, so I won't be able to do this. The workshop enrollment process is also the same. You simply have to go on any workshop, select the workshop you like. You can see the coursework, you can see the sessions that are covered there. and you have to click on Enroll Now button. And you will be able to see the pricing, and then you have to click on Buy Now button. Workshops are comparatively cheaper to the training program, so you can buy that. And now moving on towards the courses. So for all the courses that are present on OmicsLogic portal, you can simply purchase a subscription as well. So there is a subscription option as well. Subscription will be on monthly basis. So all the courses that are present on the OmicsLogic portal, we have 30 plus courses. So you can purchase those courses using our subscription model as well. So registration process for this is also the same. You have to enter your details, select a payment method, and you can get yourself enrolled. Now, if you have any questions, the stage is open for you all. You can paste your queries in chat box and let me know if you're facing any difficulty